Okay, before we begin, can somebody please confirm you can hear me?
I can hear you.
I can hear you.
Thank you. Are there any questions? Don't forget you need to choose a infectious disease project and get it cleared with me before you begin the project. A large number of students are finding that I'm saying you got to narrow it down. They're taking a topic like pneumonia, and you can't write it on pneumonia. There's too many agents that cause pneumonia. What else do I need? All right, let me share my screen. I did some grading over the weekend and last night. So I got one of the labs graded. I don't remember which one that was, and I don't want to show it now because I'm sharing my screen. Suppose I'm going to stop sharing my screen. And let's take a look at the class. Oops, I just logged out of Canvas. That was a mistake. Let me see if I can get it back. Nope. They have a new system for getting into Canvas, and it actually requires me to make 11 clicks before I can access the class. Come on, Grace Clark. Oh, I'm going to have to do it again, probably. Yeah, I can show you the start of this. Come on, share the screen. I'm already in 2 clicks, so let's share the screen. So once I come here, I have to go there. And then I have to open up my, oh, that worked. Oh, that's weird. Usually it requires me to verify my existence and require an email, but I just did it. So maybe it knows I just did it. All right, let me stop sharing the screen. I'll call up the grade book. I got the last lecture worksheet graded. And I think I got one. The practice quizzes are, well, I thought the practice quizzes are graded, but it looks like just practice quiz 00A. Let me look into practice quiz 00B and see what's going on here. That's the wrong place. Yeah, practice quiz 00B is not graded. I thought I had done that also. And then one of the extra credit study assignments is graded. I did give a few of you a warning saying you need to do something. Mostly people got warnings. You need to quiz students in your study group. The extra credit study sheet is graded. And then worksheet for lecture 2 is graded. Oh, shoot. When did that happen? It was in the grade book, and then I just left the grade book for some reason, which wasn't anything I did. Hopefully you're not seeing this. Worksheet lab 2 is graded. You have until this Saturday to get it. Corrections, if you want full credit, it'll be for full credit. And then worksheet lab 3, I did not get graded. I doubt I'll get it graded by tonight. If I do not get it graded by tonight, I'll extend the deadline, not this Saturday, but a week from this Saturday. There is a slight chance I might get it all graded by this tonight, but I doubt it. All right, any questions about any of that?
I
Let's
do
go.
have a question, not about that. In the grade book, it shows two quizzes due for tomorrow, but not in the modules, it only shows one. Do we have one or two due tomorrow?
I believe just one. Let me
It
see.
shows 0B, 01B, and C. Like there's two due tomorrow, 75 point and 51 point. But it doesn't show in the
Where are
mod.
you saying only in the grade book that's showing?
Yeah.
Okay, take a look only on the Canvas page. I'm not sure why there are two showing in the grade book. It should be, I think, quiz 1C.
Yeah, in the modules it only shows one, but I just want to make sure the other one was going to pop up on me
No,
that
the
morning.
other one, probably a year ago, there was a student who requested a different exam, and probably I made that available to that student. And apparently, I didn't realize that's in the grade book. I'll have to go look at that and remove it from the grade book. I don't even see it here. Let me share my screen again. On the modules, it's not there, and so I didn't notice that. But there's only one quiz,
Okay, thank you.
75 points. All right, let's go to the syllabus. So today is April 23rd, and we're supposed to finish chapter 2. Hopefully we'll do that, and then start chapter 3. And then, yes, quiz 1 is due tomorrow, 11:59 p.m. tomorrow. Quiz 1 covers chapter 1, chapter 10, and only the start of chapter 2, what we covered last week. It also covers labs 0, 1, and 2. Any question about any of that? All right. Let's go to chapter 2. I have that we had talked about the 4 levels of protein structure, and I believe you watched the movie. So let me state that there's 4, the primary level, the secondary level, the tertiary level, and the quaternary level. The first level, the primary level, is just the specific sequence of amino acids in the protein or polypeptide chain. So the orange one's followed by the blue one, followed by the yellow one, the sequence of amino acids. And when the protein is first made, all it has is the primary structure. But shortly after the protein is made, nearby amino acids start interacting with each other. And we call these localized interactions because it's only the nearby amino acids which start interacting with each other. Like between this one and that one, it's showing a hydrogen bond forms between these two amino acids. That could be either an ionic bond or a hydrogen. The localized interactions are amino acids that are neighboring, and they can have different types of interactions, hydrogen bonds, ionic bonds, that'll be attraction. There can be hydrophobic interactions, like a hydrophobic likes to be around another hydrophobic amino acid. There's also repulsions that can happen, like maybe between these two. This could be amino acid with a positive charge, and that could be an amino acid with a positive charge. So they will be repulsed from each other. This could be a polar amino acid, and this could be a nonpolar amino acid. They will be repulsed from each other. And I think that's all of the the localized interactions I need to talk about. Hydrogen bonds is the main one, meaning that's the one that causes most of the interactions. But in the movie, it only mentioned hydrogen bonds. It isn't true. Ionic bonds can form between amino acids. If this one is positive charge, that one is negatively charged, there will be an ionic attraction between them. And if this is positively charged and that's positively charged, that'll be an ionic repulsion between those two. That can happen. Hydrogen bonds, of course, will be attraction. And then you can have repulsion between a polar amino acid and a nonpolar amino acid. And then very rarely, it's not shown here, you can have attraction between two nonpolar groups. We're not going to talk about that much, but it's called van der Waals forces. And actually, why liquid oil is a liquid is because the oil molecules are attracted to each other with van der Waals forces. They're a very weak attraction, and they make the oil a liquid. If you didn't have that attraction between the molecules, you'd have gaseous oil. Any questions about any of that? All right. So there's a large number of secondary attractions and repulsions. They do make two major twisting or folding of the polypeptide chains. There's actually a third that wasn't mentioned in the movie, but you can have the alpha helix shown here. You can have the pleated sheet shown here. And the pleated sheet is actually the amino acids are going this way, and there's hydrogen bonds forming between the amino acids in the pleated sheet. And then the third I'll show you, it wasn't mentioned in the movie. Let me get paint open. It's called the random coil, where the amino acid just coils randomly. And there can be attraction in this region here, attraction here, and then maybe repulsion here. And we'll say repulsion there too. And obviously there'd be repulsion here. Because if there was attraction, that would be binding. And that's just a random coil. After the secondary level forms, the amino acids from different regions of the protein can start interacting with each other. And this will make the three-dimensional structure of the protein. You can call this, when the protein coils, the supercoiling will be the tertiary structure. That'll be interaction between amino acids all over the protein. For example, this amino acid here is being attracted to this amino acid here. This is one of the last amino acids in the protein, and this is an early amino acid in the protein. You can find all of the secondary interactions between amino acids in the tertiary interactions, meaning there's hydrogen bonds, hydrophobic interactions, ionic bonds for attraction, and then there's repulsion between a polar group and a nonpolar group. There's repulsion between a positive ionic charge and another positive ionic charge, or two negative ionic charges. That would be repulsion. And there can be hydrophobic interactions as well. And let's see, what did I say? The repulsion between a polar region, polar amino acid, and a nonpolar amino acid. So all of those can be seen, and all of those are seen in the secondary structure. You see them all again in the tertiary structure. There is one other interaction between the amino acids that happen only in the tertiary structure, and that is the disulfide bonds. let me blow this up so you can see it better. Between cysteine amino acid here and a cysteine amino acid here, cysteine has sulfhydryl groups. Let me show you that in paint, I think. So we have an amino acid going like this, an amino acid going like that, and there can be a cysteine here and a cysteine here. Let me get text here. So cysteine, oops, has a sulfhydryl group. And then there's a cysteine here. Let's go ahead and just put it in the same thing. Another sulfhydryl group connected to a cysteine. A little far away. It should be a little closer here, but that's all right. Well, this hydrogen can come off, and that hydrogen can come off. Let me erase that. And then the sulfurs can be linked together. Oops, wrong one. Ah, with a disulfide bond. About there. Meaning this sulfur from the cysteine is directly connected to the sulfur from the cysteine. And that is a covalent chemical bond. That is a very strong interaction. How strong is this interaction? When you get your hair perm, what they do is they make disulfide bridges between two sulfur and cysteine to make your hair the perm. And that will allow your hair to be perm for about a month, even when you go into the shower. That's a very strong bond. It does not break easily. Whereas if you were to curl your hair, my mother used to do this. She would get her hair in the curling iron or whatever. She'd first wrap her hair up to make the curl in a curl and put a bobby pin in and then put the cap on and then steam the hair to get it to curl. And then she would take it out and remove the whatever you put the hair around and the bobby pin. And then the hair would form a nice wave. That was actually only forming hydrogen bonds between the hair. And then she would go into the Western Oregon air, which would frequently be mist or rain, and then that curl would just sag because that was only a hydrogen bond. Any question about that? The point is this disulfide bridge in the tertiary structure is a very stable and very strong interaction. Any questions about any of that? All right. Now, for many proteins, in fact, I would say all enzymes that I know of, and many other proteins, they only go up to a tertiary structure. And that's because only one polypeptide chain makes the protein. So that would be something like catalase or catecholase or any other enzyme you've ever heard of. There's usually only one polypeptide that makes the enzyme. So that protein can only have a tertiary structure. But if there's more than one polypeptides that come together to form the finished protein, then we can have a quaternary structure. And that is when, let me see if I can get that out of here. Let me just end the show and go backwards. When the four polypeptides come together, they will have interactions between the amino acids on the different polypeptides. And the final protein shape will then form. All of the bonds that you saw in the tertiary structure can be in the quaternary structure. And this includes hydrogen bonds, other weak forces, as well as the disulfide bridges, which are very strong bonds. There are some proteins like hemoglobin, insulin, and antibodies, which have a quaternary structure because they are formed by more than one polypeptide chain. Hemoglobin and antibodies are formed by 4 polypeptides that come together to make the finished protein. Any questions about the different levels of protein structure? All right. Proteins can have an overall shape, and we say if the protein is roughly compact and roughly spherical, that it is a globular protein, such as hemoglobin. If, on the other hand, the protein is thread-like or fibrous, we call it a fibrous protein. And then collagen and some of the other structural proteins are fibrous proteins. Proteins can also conjugate with other molecules. So a conjugated protein is a combination of amino acids with other organic or inorganic components. This protein is usually named by their non-protein component. Glycoproteins are a protein combined with a glycogen, which is a carbohydrate. And then lipoproteins are proteins combined with a lipid. And you can find both of these in your blood as well as on the cell surface, meaning in the cell membrane, lipoproteins and glycoproteins. Any questions about conjugated proteins? All right. When we're talking about proteins, you can get protein denaturation. This is when the protein loses its three-dimensional shape. It can happen when it breaks hydrogen bonds, or perhaps forms hydrogen bonds that shouldn't be formed, and then break other bonds as well. The protein will no longer function when it is completely denatured. A protein can be natured when it is at the wrong temperature, the wrong pH, or the wrong salt concentration. So all proteins have to have the correct temperature, the correct pH, and the correct salt concentration for the protein to be in its correct shape and then to function at its optimal level. Any question about that? And when it's in the wrong environment, the protein will start losing its shape and will start losing its ability to function. And then we say it is becoming denatured. And if it's totally denatured, you just say it is denatured. An example of a denatured protein is when you take egg whites and crack the egg white is actually kind of clear and runny in a liquid. And when we heat it, we denature the proteins, changing the color to white and then changing it from a liquid to a solid. Any question about any of that?
Uh, oh, sorry. I have a
Well,
question.
go ahead.
Um, so for the quiz tomorrow, is there going to be any like organic compounds on the quiz, or is it just going to be talking about inorganic, like water and pH and stuff?
I believe I said quiz 2, chapter 2 is on the quiz.
So all of chapter.
Oh, it just says quiz 1. Oh, it's only the start of chapter 2. So,
Okay, so like up till
no, yes, the what we covered last week. Well, let me think about that. What we covered last week will be on the quiz.
Okay.
Um,
So I'm just clarifying like the stuff that like we're talking about now, like the lipids, that's not going to be on the
the
quiz.
lipids, the
But water,
carbohydrates, and the proteins will not be on quiz one. But
but
if
the
you're talking about,
water
yeah,
and like
water will be, and inorganic and organic compounds will be.
okay. Okay. Sounds good. Thank you.
All right, let's move on to nucleic acids. There are two nucleic acids you need to know, DNA and RNA. DNA is an abbreviation for deoxyribonucleic acid, and RNA an abbreviation for ribonucleic acid. You may learn either name, by the way. These molecules consist of monomers, or subunits, that we call nucleotides, and a nucleotide itself consists of three different parts. A 5-carbon pentose sugar, it's deoxyribose in DNA, and ribose in RNA. A phosphate group, so there's the sugar, there's the phosphate group, and then a nitrogen-containing base, and that's here or here. And it can be a purine, shown here, or it could be a pyrimidine, shown here. You don't need to know that now, but we'll talk about those in the next slide or the next two slides. Uh, the nitrogen containing bases in DNA, there are 4, and actually in RNA, there's also 4. In DNA, they are adenine or A, guanine or G, cytosine or C, or thymine or T. And you may learn either the name or the abbreviation. I don't care which. The purines, adenine and guanine, lack a Y, and then the pyrimidines, cytosine and thymine, contain a Y. And this is how you can remember which ones are pyrimidines, because pyrimidines has a Y in it, and so does cytosine and thymine. Any question about any of that? The purines do not have a Y in them. Now, for RNA, they have um, 4 nitrogen-containing bases. 3 of them are the same. The adenine, the guanine, and the cytosine are the same. However, thymine is replaced in RNA. It's only seen in DNA, and it's replaced by uracil. Uracil replaces T or thymine in RNA, and thymine is a pyrimidine. And thymine has a Y in it, so it's a pyrimidine. Uracil, you just remember it replaces thymine, and so it is a pyrimidine as well. The complementary bases pair to each other, they form hydrogen bonds. And so A and T in DNA will come next to each other and will pair, forming hydrogen bonds. And then the C and the G will come next to each other and pair and form hydrogen bonds. And obviously that could be T on this side and A over there, and G here and C there. The A does not pair with the C or the G. It is not complementary to a C or a G. The A is only complementary with the T. And so on one strand of DNA, if you have an A, you know on the other strand you'll have a T. And that's what we mean by complementary base pairing. Any question about any of that? How the complementary base pairing happens is the A and the T hydrogen bond to each other, and the C and the G hydrogen bond to each other. The A and the T have 2 hydrogen bonds between them. You don't need to know exactly where, but it's the nitrogen here. hydrogen, forming a hydrogen bond, well, actually with that hydrogen of this thymine. And then this hydrogen of nitrogen here is forming a hydrogen bond with this oxygen of thymine. So the important part is the A and the T form 2 hydrogen bonds. The C and the G form 3 hydrogen bonds. And I don't really have a good picture of that, but you can see it here if I blow this up. The C and the G form 3 hydrogen bonds between them. Any question about any of that? So in DNA, the sugar is deoxyribose. Might move that down here. Deoxyribose right there. The DNA is normally double-stranded. So in our cells, the DNA is normally double-stranded. However, we do have single-stranded DNA in our cells at certain times. Whenever the DNA is being replicated or transcribed, the DNA will be single-stranded. The DNA is hooked together, meaning one single strand of DNA. Come on, mouse. Is hooked together by the sugar phosphate backbone, meaning here we have a phosphate, there's the sugar, there's the phosphate, there's the sugar, and that's the backbone of the DNA single strand, holding the DNA together by a covalent chemical bond. The DNA is double-stranded, normally, but it is only held together by hydrogen bonds. Hydrogen bonds in the temperature of our body break and reform all the time. So this bond here can break, but the DNA will be held together by that bond and these bonds and all the other bonds. And then that one can reform, and then this one can break. We call that actually the DNA breathing, where the hydrogen bonds break and then reform. It's just a term. DNA forms a twisted ladder in the double helix, and the bases do not hold the single strand of DNA together. The bases are on the steps of the ladder, meaning they're off on the side, and then they hydrogen bond to each other to make the complete step of the ladder. Any question about that? Let me end the show here. RNA, the sugar is ribose. RNA is single-stranded. All of our RNA is single-stranded. The ribonucleic acid is held together by covalent chemical bonds between the sugar and the phosphate. So we say the sugar and the phosphate hold the RNA strand together. The bases are off on the side. They do not hold the single strand of RNA together. Because RNA is single-stranded, these bases do not pair with something, usually. There are times when the base can pair. We'll talk a little bit when we're talking about RNA transcription. This RNA base can actually pair to the DNA. But normally RNA, the bases do not pair because RNA is single-stranded. There are three major kinds of RNA in cells: transfer RNA, called tRNA, messenger RNA, also called mRNA, and ribosomal RNA, called rRNA. What is the difference between tRNA, messenger RNA, and rRNA? Do they have any difference in their sugar? That's a yes or no question, people. Think about it. All right, we'll move on. Do they have any difference in their bases? Does tRNA have a different base than messenger RNA? That's also a yes or no question, people. Think about it. And nobody's going to take a guess, I take, for what's the difference between tRNA, messenger RNA, or rRNA? And that may be a quiz question, so you might want to think about that. All right. ATP is another ribonucleic acid or yeah, it's a component of the ribonucleic acid. ATP is important in cells because it is the primary energy source for most cellular activities, meaning if a cell needs energy to perform a function, the cell will most likely get that energy from ATP. ATP has ribose, adenine, that's why it's called ATP, and then 3-phosphate. The T stands for adenosine triphosphate. T is the tri. And what the cell does is it breaks off either this bond, or maybe even both of them to get energy, meaning this bond here as well as that bond there is a high energy bond. If the cell wants energy, it can break that bond, releasing the phosphate, and that releases a lot of energy, which the cell can use for its energy needs. If the cell wants more energy, it can then break off this bond as well. And that's why ATP is called the cellular currency or the energy currency of cells. Because if the cell wants energy, it usually gets it from ATP. ATP is made by dehydration synthesis. You get ADP, adenosine diphosphate. You add on inorganic phosphate, that's what the I stands for, just count it a phosphate. You add energy, and then you make water and ATP. So the reaction goes this way to make ATP in dehydration synthesis. And then ATP can be broken by hydrolysis, breaking off the third phosphate group, releasing the energy for the cell's energy needs. You just add water, break off the phosphate, and that releases the energy. Any question about any of that? So cells constantly break down ATP to get their energy needs, and then they constantly make ATP so they can store their energy in ATP. All right, any questions about any of that? If not, what molecules have we discussed, both today and anywhere in chapter 2, that have hydrogen bonds that give the molecule important properties? What molecule has hydrogen bonds?
Water.
Yes, water has hydrogen bonds. Sorry, here, I got my mouth full. And what would happen to water if all the hydrogen bonds were to disappear? What would happen to water if all the hydrogen bonds were to disappear?
Would it evaporate?
Yeah, water would evaporate. You'd have no liquid water. And if you had ice, you'd have no ice. It would become gaseous water. So you couldn't have life if you didn't have liquid water. And then DNA has hydrogen bonds, as well as proteins and carbohydrates. And these molecules will not function properly, if at all, if all the hydrogen bonds were to break, simply disappear. For example, your DNA wouldn't be double-stranded if all the hydrogen bonds were to disappear. Just something to think about. All right, any questions about chapter 2? If not, let's end chapter 2 and begin chapter 3. As is normal, I give you an introductory slide giving you the major goals and a rough outline for the chapter. So as always, know the major terms, the major goals. For all of the terms, you should really go to the objectives. But know what resolution is, refraction, what are stains, what's a basic and acidic dye, what is a smear preparation, what is a mordant, and what is peptidoglycan. Let me mention that when I give a quiz question on what is a bacterial smear, every term, at least one student has gotten that wrong. Okay? So you're forewarned that every term I've always had the student getting something wrong about a bacterial smear. All right, 2, understand the sizes of organisms and the properties of microscopes. Understand the major differences between a light microscope and an electron microscope. 3, understand why staining improves resolution, and you should know the 3 different types of stains: a simple stain, a differential stain, and a special stain. And then 3B, understand the gram stain, its procedure, and the basis of its differential staining. Any questions about any of this? All right. In chapter 3, you have to know the metric system. The metric system is a unit of measurement which is used by most countries in the world. It's actually a very simple system. It's based on a multiple of 10. Like, let's blow this up. So 1 meter is the basic unit of a length. A decimeter is 1/10 of a meter. A centimeter is 1/10 of a decimeter, or 1/100th of a meter. A millimeter is 1/1000th of a meter, or 1/10th of a centimeter. A micrometer is 1/1,000,000th, or 1 times 10 to the 6 meters. 1 times 10 to the 6 over a -6 meters. And this is the size that we see under the light microscope, a micrometer. A nanometer is 1/1000th, or 1 times 10 to the -9th meters. And this is the size you see under the electron microscope. Come on, mouse. You don't really need to know a picometer, but that's 1 times 10 to the 12th, -12 meters. And I forget what that's called, though. Obviously beyond a billion. And then there is something where there's 10 meters. I think it's called a decameter. And then there's something called the 100 meters, and that's a hectometer. You don't need to know those terms, but you should know a kilometer, and that's 1,000 meters. So it's very easy converting from metric to metric because everything's based on a unit of 10. You just need to know the different orders. And like I said, if you know a nanometer, a micrometer, a millimeter, a centimeter, a meter, you don't need to know a decimeter. And then a kilometer, you should know what those are. And then if you're not using length, meaning you're measuring something other than length, like you're measuring volume, you would use a liter instead of a meter, and then you use the same prefix. So there's a kilo, liter, and a centiliter. And then if you're measuring weight or mass, it is gram is the basic unit. And then you use a kilogram or the centigram to measure uh, the weight or the mass. And let me see if I can remember this right. Weight is something you have on the planet Earth. Mass you always have, like in outer space where you weigh nothing because there's no gravity, but you still have mass. And so on the planet Earth, it would be the same, but like in outer space, it's not the weight; it would be the mass. Any question about any of that? You do not need to know the conversion between metric and English, and that's very difficult. I do remember that 1 inch equals 2.5 centimeters, but that's the only thing. Well, I do know that 1 meter is very close to 1 yard. I don't remember which one is larger, but they're like within 3 inches of each other. Okay, any question? You are expected to know the metric system on quizzes and exams. And here's one reason why you should know the metric system. You are looking at all the countries in the world that do not use the metric system. And as you can see, there's only 3, and 2 of them are converting to using the metric system. President Jefferson, meaning the 3rd president of the United States, passed a law to get the United States to slowly start using the metric system. And if you notice, we are starting to go that way. Like on the rulers, we do have metric on them. And on all cars nowadays, they have the metric system in them, as well as many signs that tell you to drive, I don't know, 60 miles per hour. It'll also have in smaller print usually how many kilometers per hour to drive. Any question about any of that? All right. And then the other reason for knowing the metric system, let's go back a bit, is because science and medicine use the metric system. Come on. There we go. So science and medicine use the metric system. So you'll run into both the metric system whenever you're dealing with science or medicine. And I think most of you know when you go to the doctors, they'll give you so many cc's of a injection. Any question about that? All right. So you should know the typical sizes of microorganisms. You should know a eukaryotic cell is much larger than a prokaryote, and it's about 10 times the size. We're talking about the average cell because the smallest eukaryote is actually smaller than the largest prokaryote cell. And then the smallest prokaryote cell is about the same size as the largest virus. But once again, eukaryote about 10 times larger than a prokaryote, and then a prokaryote on average about 10 times larger than the average virus. Although viruses do have a very wide range of what size they are, wider than a prokaryote and a eukaryote. Well, maybe not wider than a eukaryote. Let me think about that. Maybe not wider than a eukaryote, because an egg is a eukaryote cell and it's pretty large. In fact, it's larger than 500 micrometers. All right, any question about that? We'll talk a little bit more about viruses, but they can be from about 20 nanometers to, for diameter, 440 nanometers in size, but they can be longer, like Ebola is one of the longest viruses, but it's really skinny. And so it's not the largest virus in diameter, but it is the longest virus. And it gets close to 1 micrometer in length. Any question about any of that? You should know the relative sizes of things, and I've already shown you this slide. You should know a person is bigger than an egg. A chicken egg is bigger than a, excuse me, frog egg. A frog egg's about the smallest thing our human eyes can see. There are a few things that are smaller, but not much. Most eukaryote cells you cannot see with the human eye. You need a light microscope to see it. And the same thing with all prokaryote cells. You need a light microscope to see it. It's smaller than a eukaryote. Um, the smallest prokaryote is about the limit that we can see with the light microscope. Below the smallest bacteria, we have viruses. Below, smaller than viruses, we have large molecules like proteins. Smaller than large molecules are small molecules like water. Smaller than small molecules like water are atoms. And that's about as far down as you can see with an electron microscope. The light microscope, you need to know the parts of the microscope shown here. This is a compound light microscope. A compound microscope is one that has two or more lenses that magnify. So we have an ocular lens that magnifies and an objective lens that magnifies. The total magnification is the objective lens times the ocular lens. The objective lens, normally on a microscope, at least all the microscopes we have in, I should say all the compound microscopes we have in the microbiology lab at Clark, have 4 different objective lenses. They have a 4x lens, a 10x lens, 40x lens, and a 100x lens. The 4x lens can magnify 4 times. The 10x 10 times. And that's what the 4x stands for. I like to call them this rather than that's the scanning objective lens. That's the low power, and you note the low power is not the lowest. That's the high power lens, and the 40x is not the highest, and this one's called the oil immersion lens. The total magnification will then be the objective lens times the ocular lens. For all of the microscopes we have in microbiology, we only have the 10x lens in the ocular lens. But the best and most expensive microscopes have a 20x lens in the ocular lens. You cannot change the ocular lens, usually. I suppose you could take out the eyepiece and put it in with another eyepiece, but that's not normally done. Excuse me here. After sneezing, now my nose started running. All right, so there's the ocular lens. You should know the tube, because or the arm, you carry the microscope by the tube or the arm. You should know the stage. That's where you put the slide. You should know the condenser. Let me blow this up so you can see that better. The condenser and then the iris diaphragm. They're always together, although in some microscopes, the iris diaphragm is above the condenser. The illuminator or light source here, the coarse adjustment focusing, that's the large knob here, the fine focusing knob, the small knob. Let me end the show there now. This is a simple microscope. It's actually the microscope that Antony van Leeuwenhoek made. It only has one lens to magnify. A simple microscope is nothing more than a super duper magnifying lens. Resolution is the ability of a lens to distinguish 2 points as 2 separate points. A microscope with a resolving power of 0.2 micrometers can distinguish between 2 points that are equal to or greater than 0.2 micrometers apart. If the two objects are within or less than 0.2 micrometers apart, you would not see them as two distinct points. The resolution limit of white light is approximately 0.2 micrometers. And that is the best resolution you can get with white light, 0.2 micrometers. Any 2 points less than 0.2 micrometers apart cannot be distinguished as 2 separate points. And that's the limit of white light. It has to do with the wavelength of light. And anything smaller than this or any 2 points closer than this, you cannot distinguish them as separate points. The best compound light microscope we have has a total magnification of 2,000 times. That is a 20x objective, and the 100x, excuse me, a 20x ocular lens and a 100x objective lens. And that will magnify and get the resolution down to 0.2 micrometers. If you were to use higher magnification, it would be empty magnification because the resolving limit of white light is 0.2 micrometers. Any question about any of that? So let me show you. Here are 2 points that are separated by 0.2 micrometers, that's 200 nanometers, and you can see them as 2 separate points. This is what they really are, but that's how they will look under the microscope. And these points here cannot be resolved as separate points or separate objects because they're less than 200 nanometers away from each other. Any question about any of that? And so that would be the resolution limit of this microscope and actually all microscopes that use white light. Any questions about any of that? All right. Another property of light is light can refract. The refractive index is the light bending ability of a medium through which light passes. When we look at this straw here, it looks like the pencil is bent right there. So this is sort of the artist's rendition. That is a straight pencil. The difference is light has a different refraction when it's going through water than at air. And so the light, when it is coming through the water and then switching to the air at the interface, light will refract and will bend. And that's why the pencil looks bent right there. Any questions about refraction? Refraction is actually very important for life. Like if a fish, excuse me, a bird sees a fish in the water, because of the refraction of light, the image seen by the bird will appear to be here, even though the fish is really here. And birds are evolutionarily programmed to know that if they see the fish here, they know to put their feet down to catch the fish here because of the refraction of light. Any question about any of that? Refraction of light is important in a microscope because when the light travels through the glass and then travels through the air at the interface of the glass and the air, light will bend, and at least some of the light will bend, and that we call refraction. The advantage of using oil is oil has a refractive index the same as the glass. And as you can see here, this ray of light is bent so hard, it does not even come out of the glass. This ray of light is bent hard enough that it will not be collected by the objective lens. That one is bent, but it will be collected by the objective lens. You'll notice it's bent once again when the light enters the glass of the objective lens. That ray of light is not bent, meaning it is not refract, and some of the rays are just refracted and others are not. And so that one will not be bent and it will be collected by the objective lens. If you put in oil there, you remove the refraction of light between the interface of the glass and the oil. And so that light ray will not be bent, but it will not be collected by the objective lens. But then this one will be collected by the objective lens. And without using the oil, it's not collected by the objective lens. And that one is collected, and that one is collected by the objective lens when we're using oil. The point is you collect more light when you're using oil than when you're not using oil because of the refraction of light. And because you're collecting more light, you have a greater resolution when you use oil. And so your object will appear better and you can actually magnify further if your resolution is better. Any question about any of that? That's why oil is used with the light microscope, because it allows the objective lens to collect more light. It removes the refraction of light, or at least the refraction of light leaving the glass. As well as it removes the refraction of light moving from the oil to the glass. All right, any questions about refraction? All right, let's talk a little bit about bright field illumination. Bright field illumination is what normally we have in a light microscope. All of the light microscopes we have in the microbiology lab are compound light microscopes, and they work by bright field illumination, meaning the light leaves the light source, goes through the specimen, and then is collected by the objective lens to be seen by the human eye. That makes the object fairly bright from the light coming through the specimen, but less bright than the background of the slide, which looks white because all we're seeing is the light from the lamp. Any question about bright field illumination? That's in contrast to dark field illumination, which you must know the concept for the quizzes. You don't know to know exactly what's going on. But in dark field illumination, let me blow this up so you can see it better. We have an opaque disk between the light and the specimen so that any rays directly coming from the lamp to the specimen are blocked by the disk. Come on, mouse. And that's why the background of the slide in dark field illumination is black, because no light is directly coming from the lamp. However, we have some light coming at an angle going through the specimen that is refracted by the specimen, and some of that light is refracted at an angle, come on, mouse, so that it is bent and collected by the objective lens. If you didn't have any refraction of the light, none of the light would be collected by the objective lens. So it's called dark field illumination because the background of the slide is totally dark, and then the specimen is darker than bright field, the specimen in bright field illumination, and that's because there's less light coming through the specimen. We only have the rays at a steep angle coming through the specimen, and then the specimen refracts that light into the objective lens, and there's less light from the specimen coming to the objective lens than in bright field illumination. So that's dark field illumination. Any question about any of that? You can notice that you can observe the specimen similar in both, even though they're very different. The most widely known seen difference is the background. This one is light, this one is dark. Any questions about bright field and dark field illumination? All right, let's move on to an electron microscope. You're seeing an electron microscope, and you can see they're much bigger than a light microscope. An electron microscope uses a beam of electrons to observe the specimen, and a beam of electrons can magnify the specimen much greater than a ray of light can be magnified and magnify the specimen. The beam of electrons is focused by an electromagnetic lens. So this is not a glass lens to focus light. It's an electromagnetic lens to focus the beam of electrons. Any question about any of that? Electrons have a shorter wavelength than light, and that's why we can magnify much greater with a electron microscope than with a light microscope. The shorter wavelength of electrons give a greater resolution, about 100,000 times greater. So this allows the examination of much smaller objects, such as a virus, the internal cell structures, shown here, or cell surfaces, shown here. There are two main types of electron microscopes. There are other types, but we're only going to talk specifically about two, and you'll be only quizzed on these two. There's transmission electron microscopy, shown here. Come on, mouse. And then scanning electron microscopy, shown here. What do you notice about the difference between these two types of electron microscopes? Come on, don't be shy. There are two very easy differences. Nobody's going to say anything, huh? I should not tell you the answer because this is a quiz question.
Is it one of them you see through? the virus?
Not really seeing through, but you are seeing a difference, and that is in the transmission electron microscope, you're only seeing the specimen in 2 dimensions. You're seeing one plane here. Come on. One plane through the cell, so you only see 2 dimensions. In scanning electron microscopy, you see 3 dimensions, meaning you see both the X and the Y axis as well as the Z axis. And you can see this cell, you can see the surface of the cell, and it is in 3 dimensions. Come on, mouse. Another difference is this scanning electron microscope is only viewing the surface, whereas with transmission electron microscopy, you can see inside the cell, and you can focus on different, I guess, layers through the cell. Any question about any of that? All right. This is a scanning electron microscope. We're seeing the crystal in 3 dimensions, and you notice we're only seeing the outside of the crystal. You're not seeing inside. And this is transmission electron microscopy. I think this is the membrane of a chloroplast, and you can see inside the cell structure here. Here, but once again, we're only seeing 2 dimensions, not the surface, not in 3D. All right, there are 2 other types of electron microscopy. You're not going to be quizzed on them, so you don't need to learn this, I guess. I'm just going to mention there's scanning, tunneling microscopy. A third type of electron microscope. It uses a metal probe to scan a specimen. It can give a resolution 1/100th the size of an atom. So much smaller than an atom. And here we're looking at, what is this? Sorry, I just covered it up. We got to move that. A protein from E. coli. There we go. For some reason that didn't work the first time I did it. This is an atomic force microscopy. You're also not examined on this. A fourth type of electron microscope uses a metal and diamond probe inserted into the specimen. It produces a 3-dimensional image of what you're looking at. And here we're looking at the toxin for Clostridium perfringens. All right, so getting back to the light microscope, here we're looking at 2 different cells, cells on the left and cells on the right. Which cell is easier to see? Cells on the left or the cells on the right?
Also on the left?
Cells on the left, this one here, are easier to see?
No, the other side.
Yeah, the
On the
cells
right.
on the right are easier to see. The only difference between these cells and these cells are these cells are stained. These cells are not. And the main reason why staining improves the resolution and the clarity is it increases the contrast between the background of the cell and the cell itself, meaning the cell here doesn't have much contrast with the background. And here, the cell has a large contrast with the background of the slide. And that's the main thing that using a dye or stain does. It increases the contrast between the cell and the background, making the cell easier to view. So with bright field microscopes, most microbes are colorless. They have little contrast with the slide or the medium. And then to see them well, you really need to apply a stain. Staining is just coloring the specimen or the background of the slide to increase the visibility and the contrast with the specimen and its background. So staining colors the specimen or it colors the background to increase the contrast between the specimen and its background. A smear is a thin film of solution of microbes placed on a slide. Ideally, when you're making a smear, you want the smear to be only a single cell layer thick. You make a smear prior to staining the microbes. Like if we were going to stain bacteria, we make a bacterial smear where the microbes are put on the smear and they're usually fixed to the slide to attach them as well as kill them. Usually we fix the cells by air drying them to the slide and then heat fixing them to the glass of the slide. And that will prevent the cells from washing off when you're adding a liquid like a stain or you're rinsing the slide, which you do when you're staining the cells. But the smear is something that's done before you stain the cells. Any question about any of that? All right, let's talk about a stain. A stain is also called a dye. It is a salt consisting of a positive or a negative ion. In the dye, one of the ions, either the positive or the negative one, is colored, and that we call the chromophore. A basic dye has the chromophore, the cation, or the positive portion of the salt. In an acidic dye, the chromophore is the anion, or the negative part of the salt. Any question about any of that? If not, I think I'll stop here. We'll continue with this next time. Any questions? All right, if there's no questions, I'll see you at 6:30 for the lab. Bye.